Hey y'all, it's Miss Welch. We're going to talk a little bit about cells and particularly the cell cycle and mitosis. So this is a cell's method of living and growing throughout its life cycle and then dividing to make new cells. So we're going to talk about the purpose and the steps of these processes, as well as maybe some fun mnemonics and a dance. Don't worry, you don't have to get up, just a finger dance to help you remember these concepts. So let's get started. Here's just a simplified schematic of a cell's life cycle. So we usually just call this the cell cycle, and that includes the entire span of the cell's life. So most of this we call interphase. That's G1, S, and G2. That's interphase, where the cell is not dividing and it's just living normal life. The M here stands for mitosis, and a lot of drawings will also have a C section for cytokinesis, although this schematic is putting that buried with mitosis as just one step. So let's look at what each of these sections means and what the cell is doing at any one point in time in its life cycle. All right, let's think about G1 first. So a cell's cycle or life cycle would start at G1, and that's just a growth phase. And a lot of times we think about this as just living the cell's normal life. So it makes me think about this like a busy city street where you see everybody walking down, everybody has a purpose, going somewhere, doing something, just living life. The cell is doing whatever it was meant to do. So if it's a heart cell, it's beating. If it's a skin cell, it's protecting you. If it's a muscle cell in your pinky, it's moving when you tell it to. So the cells are doing their functions, they're just living life. Now, at some point, the cell is going to have to make a decision on if it should divide. So think about that like a person's lifespan. Should we make another one of us? Should we have a baby? And so everybody has those life crisis moments. What should we do? And that would be the checkpoint at the division between G1 and S. Anytime you get to these lines that divide one stage from the next, so right here between G1 and S, the cell has to make a choice and it has to decide whether to move on to the next phase that's checkpoint. So the cell here is going to decide, do I need to divide? Do I need to make another cell or not? If the answer is not, then the cell will just stay in G1. Sometimes these cells actually can go into a stage that we call G0, where the cell is not actively dividing at all. This makes me think about if someone decided to become a nun and go to a convent and never have children and live alone off by themselves. Maybe that's an extreme example, but the cell is not going to divide. There's no way, no how. It doesn't even have to think about it again. There are some cells in your body that hardly ever divide, if ever, and we say they are stuck pretty much permanently in G0. Think about your neurons, your brain cells. We know that brain cells don't usually come back. If you have a head injury, if you have brain damage from drugs or from cancer, tumors, etc., they don't grow back even if they're needed. And so neurons are typically in a G0 state where they're doing their function. They are acting as neurons, but they are not going to divide. Now, there is actually some newer research that says neurons may divide about once a decade. And so we hope that that is good news for future research into helping rebuild those brain pathways and those new neurons after someone's had damage. All right, so back to interphase. If the cell does decide it's time to divide, then we'll move on into the S phase. Now, S phase also stands for synthesis, and it's where we are going to make new DNA for the cell. We're going to replicate the DNA or synthesize new DNA. So this makes me think about when a couple has finally decided it's time to move on and go to that next phase in their life and have a child. So if you're going to make that important decision, then you're going to have some preparation involved, right? You may think about where you're going to live. You may think about what jobs you have, or even smaller scale things like setting up the room in the nursery and preparing for the arrival of the child. So there's lots of things that are going to happen, a lot of preparation, and so that's that synthesis phase. In our cell, we're worried about the DNA. You can't just divide the cell in half and give each cell half the DNA, that doesn't work. The cell will not be the same. So we need to make an exact copy so that the new cell will have everything it needs genetically to create the instructions for that new cell to live by. We have another checkpoint at the end of S phase. When you get to the end of S phase here, before you go to G2, the cell checks to make sure that DNA copied correctly. You wouldn't want the DNA to be either mutated or not finished copying or any other kind of problem. So the cell does do a self-check to decide, has the DNA copied correctly? So we now have 2 copies of all the chromosomes. And if so, then we can move on to G2. G2 is the growth phase. That reminds me of just the growing and getting ready for the baby to be born, that belly bump getting bigger and bigger. For a cell, they're going to have to copy all the organelles. So just like we have to have 2 sets of DNA, you'd also want that new cell to have enough mitochondria, enough ribosomes, enough Golgi apparatus, all the different organelles that the cell will need to grow and survive. You want to make a duplicate or a bunch of duplicates of each one. That way, when the cell does divide, each cell has everything it needs to get off to a good start in life. Now, the last phase, there's a checkpoint there before we move on to mitosis. Make sure everything's ready again. The cell has a lot of built-in checks to make sure that we're ready for the next step before we move on. Then the last phase is mitosis, and that is the actual process of making the new cell and dividing that first cell into 2 identical copies. Mitosis and cytokinesis have several steps, and there's a lot of things going on at the same time. So I've pulled up this chart. It may be a little small for you to see, but I'm going to try to talk you through it and show you through it with a little finger dance to try to help you remember it just in a different way. You know, movement is a great way to remember things. There's also a lot of songs out there to help you remember this type of thing. If you're ever stuck, you can go to YouTube and type in a topic and song and see if anybody's made a good one, whether they're catchy or not. Sometimes they get stuck in your head. It helps you remember. So there is a mnemonic, though there's a lot of mnemonics for mitosis. I think the easiest one for me is I peed on the mat. See it? So I for interphase, P for prophase, mat. I peed on the mat, M-A-T, metaphase, anaphase, telophase. See it, C for cytokinesis. So you might come up with some other mnemonic yourself to remember the steps in order, but that simple phrase has always worked for me. So let's talk through this. So in interphase, all of the DNA is in the nucleus, of course, but it is uncoiled. It is loose. All the chromosomes are loosely arranged because the different proteins and enzymes and molecules all have to get in there to the DNA to cause it to work, to cause things to happen. So it can't be all tightly bound up. It has to be spread out. So in interphase, that's the case. However, when we're ready to divide and make the 2 new cells, we wouldn't want a big pile of spaghetti to try to divide in half. We want those chromosomes more compacted and easier to transfer. So in prophase, one of the first things you'll see will be all those chromosomes will condense down into individual chromosomes so you can see them. So like my fingers are going to be chromosomes in the little dance, all right? So in interphase, they're hidden. So it'd be like, oh, they're behind my back. You can't see the chromosomes. In prophase, they have condensed and you can see them. All right. And there's 2 of each, right? Because they're copied. They actually would be stuck together, but I can only do so much here. Okay. Spindle fibers, I can't do it with my fingers, but spindle fibers would move to opposite sides of the cell and the nuclear membrane would break down. So whereas in interphase you couldn't see my chromosomes because they're in the nucleus hidden by the membrane, now in prophase they are condensed and they are visible because you're breaking down the membrane to see what's inside the nucleus. Now I'm skipping prometaphase if you see that on here. You know, this process is not a one jump at a time steps. It is a continual gradual process. So it's hard to see those jumps Some books will have as many as 10 or 14 or 15 or 20 steps. They do pre and middle and pro and late phase of each of these. We're going to do just the basic core ones. So prophase. Chromosomes are visible. The nuclear envelope has dissolved. Metaphase means middle. So the chromosomes line up in the middle of the cell. So metaphase, middle. They line up along the metaphase plate. And then now the chromatids, which each of these pairs, so if they're joined together as duplicated chromosomes, the same ones just copied, they are joined in the middle by a centromere, and at this point a spindle fiber attaches to each one at that centromere in the middle. Okay, so I can't really do it with my fingers that way. We'll do it this way. They would be kind of like this, but we'll do it like this. So the metaphase, they line up in the middle. All right. Anaphase is where the chromatids separate. So the two chromatids that were together and are the same chromosome, just duplicated, they now separate. The spindle fibers are attached to each one, and it pulls them apart toward the centrioles on the opposite sides of the cell. Okay, so anaphase, the sister chromatids are split apart. Telophase is kind of the reverse of prophase. Those chromosomes are going to unwind, they're going to loosen up, and the nuclear envelope is going to reform. So we're going to see them unwinding, but we're not going to see them anymore because they're inside the nucleus, in that nuclear membrane. We can't see it. And then cytokinesis, the two new cells are made, they're completely formed. Cytokinesis is cellular or cytoplasmic division. Telophase, the nuclei form two nuclei, and then cytokinesis, we form two completely new cells. The cytoplasm divides in half. Now let me just walk it through like all the way through in one time. So interphase, chromosomes are behind my back. They're doing their thing. Prophase, chromosomes are visible, duplicated, nuclear membrane broke down. Metaphase, they line up in the middle. Anaphase, they separated. Telophase, the nuclei separate, and you see the two separate nuclei. And then cytokinesis, you see two separate new cells. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. Okay, that's as much dancing as I do. But sometimes doing it with your hands helps you remember each of those steps. So I know this is a lot of information in mitosis, a lot of parts and pieces, so go over it a couple times. If you need more help, you might practice drawing it. You might practice cutting up, printing it out or cutting out all the little pieces and facts and see if you can match them to the right phase. You could look for a song. There's, I think, a mitosis rap. There's all kinds of different things out there. But put a little time into it till you understand the concepts of all those steps put together. All right, I hope you enjoyed reviewing the cell cycle and mitosis. That's all for now. Thank you for watching.